Hello students, in previous part we have studied about different types of proteins and now in this part we will discuss on lipid. Lipid is a microbiomolecule. It is a microbiomolecule. Actually, lipids is mainly three types. Lipid is of three types. There are three types of lipids. First one, it is the simple lipid. Second one is conjugated lipid. And third one is lipid derivative, or we can say that derived lipid derivatives. So we can say that there are three types of lipids. Lipid is microbiomolecules. Simple lipid is mostly the combination of ester of fatty acid and alcohol. The simple lipid is mostly present in which is the ester of fatty acid and alcohol. On the basis of composition, simple lipid we can categorize in two parts. The simple lipid we categorize into two parts on the basis of different components. First one is true lipid. True lipid is also known as neutral lipid. This true lipid is the ester of fatty acid, and glycerol. The ester of fatty acid and glycerol, that makes the true lipids or neutral lipids. And second type of lipid is waxes. This is also the composition contains ester of fatty acid with alcohol, except then glycerol. There is no glycerol present. Ester of fatty acid and alcohol without glycerol. That is known as waxes. Firstly, we discussed on true lipids. Second one is conjugative lipid. The lipid which is formed from the simple lipid as well as non-lipid part. Conjugative lipid means it may consist of simple lipid plus non-lipid part. It is the composition. It is a combination of simple lipid. It may consist lipid and non-lipid part. That is known as conjugative lipid. For example, glycolipid, phospholipid, and lipoprotein. There are different types of conjugative lipid. Conjugated lipid is the lipid which may contain simple lipid and non-lipid part, such like as glycolipid. Glycolipid is the composition of carbohydrate plus lipid. Phospholipid, it contains phosphate and lipid. Lipoprotein, it means it contains protein and lipid. and lipid derivatives. There are found different types of lipid derivatives we will discuss in later session. For example, sterol and terpenes. We will discuss later in the next session. Firstly, we have seen that lipid is a microbiomolecules. There are three types of lipid: simple lipid, conjugated lipid, and lipid derivative. Simple lipid is mostly the composition of ester of fatty acid and alcohol mostly. But on the basis of composition, we can categorize the simple lipid in two parts: true lipids and waxes. True lipids or neutral lipid, it contains it is the ester of fatty acid and glycerol, and waxes contain it is the ester of fatty acid with alcohol without glycerol. Firstly, we will discuss on true lipids. What are true lipids and how they can be produced? True lipid. As we know that true lipid is also known as neutral lipid. Neutral lipid. It is the ester of fatty acid and glycerol. Fatty acid. Fatty acid is contain a long chain of hydrocarbon compounds with carboxylic group. It is the fatty acid means it is the long chain of hydrocarbons with carboxylic group, carboxylic functional group. For example, we can represent this is the R. R stand for hydrocarbon, long chain of hydrocarbon compounds, and it is attached with the one carboxylic functional group means COOH. This is the ester of fatty acid. We can say that it is a fatty acid, long chain of hydrocarbon compound with contain of carboxylic acid. We categorize this fatty acid on the basis of requirement, two types. On the basis of requirement, the fatty acid of two types, essential fatty acid and non-essential fatty acid. Essential fatty acid or non-essential fatty acid. Non-essential fatty acid is the fatty acid which is synthesized by our body, but there is no external requirement in our diets. This is synthesized by our body. There is no need to take in our diet. That is non-essential fatty acid. Essential fatty acid, that fatty acid which is not synthesized by our body, but we must be taking our diet. This is not synthesized by our body, but it must be, but it must be in our diet. Essential fatty acid, basically two types of what we can categorize into three. Essential fatty acid is linolenic acid. It may contains omega-6 unit. Second one is alpha-linolenic acid, and it may contains omega 3 units. Basically, two types of essential fatty acid. But as this diet, as we can say that this fatty acid becomes in our body less quantity, the first linolenic acid, it may change into third part, that is called acridonic acid. These acids are very essential for our body to perform functions. So we can say that we categorize in essential fatty acid, which is not synthesized by our body, but they must be taken in our diet. Second one, we can categorize the fats into two different parts, because we have know that fatty acid is the long chain of hydrocarbon compound. How these chains are combined with each other? On the basis of this combination of hydrocarbon compound, we categorize the fat into two parts. Fats into the two parts: saturated fat, or we can say saturated fatty acid, and unsaturated fatty acid. Saturated fatty acid and unsaturated fatty acid. Saturated fatty acid, in which hydrocarbon compounds are attached with each other by the single bond. It may contain all single bond. That is called saturated fatty acid. And if the hydrocarbon compounds are attached with each other by double or triple bond, then that is called unsaturated fatty acid. It contains double or triple bond. Then that type of fatty acid is known as unsaturated. The next one is glycerol. Glycerol is the compound. Glycerol. The chemical formula of glycerol is CH2OH, CHOH, CH2OH, and all are attached with the bond. Glycerol contains 3 hydroxyl group. It contains 3 hydroxyl functional group. As glycerol combined with fatty acid, it forms ester bond. How the glycerol combined with fatty acid? As we have discussed, simple lipids. What is simple lipid? It is the ester of fatty acid and glycerol. How these esters are produced? Glycerol, the chemical formula CH2OH, CHOH, CH2OH, and it contains 3 hydroxyl group. Suppose if the fatty acid attached with glycerol, such like this one, R, it contains COOH. When fatty acid combined with glycerol, hydroxyl, it eliminates one molecule of water. It eliminates one molecule of water, and here bond is formed. That bond is called ester bond. Ester bond. When alcoholic group of glycerol combined with fatty acid carboxylic group, it eliminates one molecule of water molecule, and that bond is known as ester bond. So we can say that there are 3 sites where we can produce ester bond. In glycerol there are 3 regions, 3 sites, because glycerol contains 3 hydroxyl group. So there are formation of 3 ester bonds can be take place in glycerol. How the structure of esters, which when glycerol attached with fatty acid, it forms ester bond, and that compound is changed into ester. A ester consists two regions or two parts. Ester consists two parts. One in circle form, that is called glycerol. This glycerol attached with the fatty acid a long tail. This is the fatty acid. The glycerol in circle is known as, it's contained as head, and in a long tail like structure fatty acid, this is the called tail. Head or glycerol is the hydrophilic. It is the hydrophilic part. What do you mean by hydrophilic? It means this is the part which reacts with the water. That is called hydrophilic. Whereas fatty acid, it is hydrophobic. It means the fatty acid tail part doesn't react with the water. That is hydrophobic. A molecule or a compound which contains two parts, which is joined with the two parts, and which are two pathway, hydrophilic and hydrophobic, that is called amphipathic. Amphipathic. So we can say that it is amphipathic. So we can say that the ester, this is the ester because glycerol attached with the fatty acid, it forms ester with ester bond. So it shows amphipathic. The amphipathic forms the glyceride. It means when glycerol attached with fatty acid, it forms the glyceride. There are three chances in which one glycerol attached with one chain of fatty acid, one glycerol attached with two chain of fatty acid, one glycerol attached with three chain of fatty acid. So we can categorize three types of glycerides: monoglyceride, diglyceride, and triglyceride. How is the monoglyceride produced? Monoglyceride contains one molecule of glycerol with one chain of fatty acid. This is called monoglyceride. If one glycerol attached with a two chain of fatty acid, then it is known as diglyceride. And when one glycerol attached with three chain of fatty acid, that is called triglyceride. There are three chains, or there are only three chains, not more than three chains. There is no chance of more than three chains combined with glycerol. So the glycerides are of three types: monoglyceride, diglyceride, and triglyceride. Glycerol present only one molecule, but the chain of fatty acid may be 1, 2, and 3. So on behalf of the fatty acid chain, we can categorize glyceride into three parts. Now we again take the fatty acid, or say we can say that neutral lipids. Neutral or true lipids. True lipid, it is also known as neutral lipid. It is categorized into two parts. Fat जो हैं, there are two types of fat: saturated or unsaturated. Saturated or unsaturated. How the saturated and unsaturated fats are present? If the glycerol with the saturated fat present, that is called saturated fat. Saturated fat always found in solid state. It is found in solid state. For example, ghee. And unsaturated fat, the glycerol when combined with the unsaturated fat, or that is called unsaturated fat. Glycerol with unsaturated fatty acid. This is also fatty acid. Unsaturated fatty acid will combine with glycerol that forms the unsaturated fat, and it is always found in liquid state. Liquid state, for example, oil. Oil is unsaturated fat and ghee is saturated fat. The unsaturated fat oil is good for health because it is easily digested. It is good for health. This is easily digested. So, as the person take the unsaturated fat inside the body, the unsaturated fat automatically change into saturated with the process of hydrogenation. It means unsaturated fat. The unsaturated fat which is made up of 2 or triple covalent bond, it changes into saturated fat by the process of hydrogenation. It means hydrogen combined with unsaturated fat and it converts into saturated. For example, this double hydrogen bond compound, when take place the hydrogenation reaction, it change into is the hydrogenation. It change into saturated. With combination of hydrogen, and that process known as hydrogenation. If a person is usually use maximum or excess quantity of unsaturated fat, this unsaturated fat automatically converts into saturated in our body. So the person is suffering from some health problems such like as cardiac arrest, and due to the deposition of, as the saturated fat is deposited in the blood vessels, so it affects the flow of blood and the person is under cardiac arrest. The saturated fat always present in animal. Saturated fat found in animals and unsaturated fat found in plants. What is the role of this fat? The role of fat is mainly 3 types. Function of fat. First, the fat which deposited under our skin and it maintains the body temperature, which acts as insulator, not lose the body heat outside and outside to inside. That is called subcutaneous fat, which is deposited under our skin and maintain body temperature. It means it acts as insulator. Second, this deposition of fat around the organs. Deposition of fat around the organs and makes a layer, and this layer protect the organs from mechanical shock. Protect, it means the fat when deposited around the organs and makes a protective layer. This protective layer protect the organ from mechanical shock. Protect the organ from mechanical shock. And third one is the reserved fat. The fat which is reserved in body act as is used in as energy source. Reserved fat uses as energy source. This is the simple fat, true lipid. The next part of simple lipid is waxes. Waxes. As we have seen, as we have studied, waxes is the ester of glycerol, fatty acid, with alcohol. There is no glycerol present. Without glycerol. That is called waxes. We can obtain different waxes such like as plant waxes. Plant wax which is known as cutin. Plant produce cutin and these wax forms the cuticle layer. Cuticle layer which protect the leaf in external environment. This is the plant wax, cutin. Bee wax, the wax which is synthesized by the bees in hives. Bee wax is soft and yellow in color. Yellow color. Firstly, the bee wax is produced or secreted by the waxy gland. Bee wax is secreted by waxy gland. These waxy glands are found the last abdominal segment of worker bee which are present in the last abdominal cavity or abdominal segment, segments of worker bee. That is called bee wax. Next is cerumen. Cerumen is also known as ear wax. This ear wax, the wax which is present in our waxy substance which is present in our ear, that is called ear wax or cerumen. This wax is produced by, or you can say produced in outer ear by auditory canal. This wax protect our ear from, or we can say that it helps in trapping the microbes as intrusions in our body and in our ears and kill them. Next one is sublim. This is secreted by the sebaceous glands which are present mostly in our face, and they secrete oil. And one more type of wax is lanolin wax. This is also known as wool wax. As we know that the hair of sheep contains special types of wax which we use as wool. So that is wool wax is known as lanolin. We not discovered one more types of wax is paraffin wax. Paraffin wax is some another features which we can obtain different substances, petrochemical substance, crude oil. So we can not categorize the paraffin wax in the simple lipid. In next part we will discuss on the conjugated lipid or we derivative of lipid, or we can say that lipid derivative.